5-minute pretest to see how their understanding on the topics of cell culture, and a short opening remarks. And I will read a curriculum vitae of Dr. Tsukada. And then we'll have 60-minute guest lecture, followed by 30 minutes of Q&A session, and then 5-minute post-test and feedback, and we will conclude our session for today. And now let's move on to the pretest. So all the students here, please scan the QR code. You will be directed to answer only 4 questions at Google Form, yeah? It's okay if you don't know the answer. For now, you will learn the answer later on the lecture. So how are you doing, Sensei? How's Chiba? How's the weather there? It's autumn, right?
It's autumn and become cold, and now today's very rain. How's yours?
Yeah, it's a perpetual summer here.
Right, I guess so.
Yeah. I still remember that autumn is the season of good foods there.
Right.
Yeah, let me check the pretest response. Oh, baru satu yang isi. Okay, I will speak in Indonesian. Silakan isi pretestnya. Silakan ya, baru yang respon 1. Bapak Ibu silakan isi sebagai syarat untuk mendapatkan sertifikat mengisi pretest dan posttest. Dan langsung tahu ya, langsung tahu skornya. Yang sudah respon langsung tahu skornya. Wah, ada yang benar semua ini. Ada yang benar 1, 2. Oke, silakan diisi ya para mahasiswa. Okay, we're still waiting for your pretest response. Oke, sambil menunggu ya. Oke, we're still waiting for the pretest response, but I think to save times, we'll move on to the next on our rundown. The next, please, Dea. Yeah, I will read. I will introduce you to our guest lecturer today, who is Dr. Tsukada. Okay, I met Dr. Tsukada maybe 14 years ago when I was a doctoral student at Biomedical University. So Dr. Tsukada at that time was an assistant professor. He's now an associate professor at the Department of Biomolecular Science at the Faculty of Science, Toho University. Of course, Dr. Tsukada has extensive research experience. As you can see here, he got his bachelor degree from Toho University, his doctoral degree from the Laboratory of Physiology, Ocean Research Institute at the University of Tokyo. And he was a research associate at the Department of Cell Biology and Anatomy, the University of Arizona. He's been here and there. Amazing. and where I met him at the Department of Anatomy, Biomedical University. And his dissertation, his passion also in ill physiology, and his expertise in endocrinology, histology, and cell biology, and members of Endocrinology Society, Pituitary Research Society, and Peptide Hormone Research in Japan. And next, there. Next slide. Okay, and Dr. Tsukada also serves as in reviewing editors of the journal Frontiers in Endocrinology. He got awards for his best papers and so many publications. You can search for him. And of course, it's an honor for us to have him to share his knowledge and insights today. Okay, Dr. Tsukada, the time is yours for 60 minutes. We can't wait to be enlightened by you.
Hi, thank you for a great introduction. And then let me share this slide first. Can you see the slide now?
Yes.
Okay, and then you can hear me.
Yes, very well.
Okay. So I'm Takehiro Tsukada from Toho University, and today I was invited by Dr. Chimi and then deliver a lecture to a student in Andalas University. And today's my presentation is the utilization of three-dimensional cell culture in biomedical research. And then Dr. Chimi already introduced me, but just let me briefly introduce myself. And then I traveling a lot of universities since this is my sixth university. And then first I'm a graduate at Toho University and almost like 25 years ago. And then I move on to our medical school to work on pharmacology and also back to our original basic biology in Ocean Research Institute at the University of Tokyo for 5 years. And then after that, I go into cell biology to its main topic at the University of Arizona. And then I was working on the embryonic stem cell work in the US. And then after that, I'm going back to Japan and then belong to, I was assistant professor at Biomedical University, where I met Jimmy-san and Reza-san and many Indonesian students. And then I was working on the rat pituitary study and basically using a histological technique. And then since 2016, I back to my own home university and then own my laboratory. And now I just stopped using my cell biology study and then now working on the ill physiology. I'm not going to talk about this topic today because, and then today's topic is cell culture. And then it's been like, I have a more than 10 years cell culture experience. And then I hope my knowledge can, be delivered to your students. And then if you have any question, even during my talk, just be free and then just put the comments or the question in the chat, or you just raise your hand and then just say your questions. Okay. So then now I'm going to start my topic, the cell culture. And does anyone do like cell culture experiment in the students or nobody?
I think they have.
They have?
I think some of them are working on cell culture. Ya, silakan yang ada pengalaman responnya, Sensei.
Okay.
They're just shy, I think.
Okay. So I just first explain the brief like what is a cell culture, and then the beauty of cell culture, and then what is the detriment of cell culture. And then the cell culture itself is one of the biological experimental technique. And then that's kind of in between animal physiology and also like biochemistry. And then this one is the cell culture. So cell culture is more, the beauty of this cell culture is you can observe simply the cellular response without any effect of hormonal or nervous control and also immune controls. So if you walking on this like animal study, so a lot of complications is occur in their body. So one is the hormones and neurons and immune cells are interact each other. So it's very hard to see the simple cellular response. And then using this cell culture, you can simplify the response and just by in a culture medium. And then it's still the live animal, the cell. So it's more physiological when you compare to more like biochemistry research. And then so this technique is very useful to just observe the cellular response. And then you can also change the cell culture environment. And then it's good for drug screenings and also modify the gene and then protein transductions using this cell culture. And it's very hard to use. It's doable to making a transgenic mice, but it's kind of a lot of efforts and monies, and just you can easily modify the gene using this cell culture technique. And then before I talk about the cell culture, how to do a cell culture, and today I'm just briefly explain how the cell culture experiment is developed. The first guy who established the cell culture is Ross Harrison in late 19, and he is an American biologist, and then he first cultured the frog neuroblast. It's a neuron cells from the frog, and then succeeded in the culture to isolate the cells from the flock, and then they succeeded in culture in lymph medium, and then he's a pioneer and then of the cell culture. And then this Alex Carrel, who is a French surgeon, is more famous because he is awarded the Nobel Prize in 1912. He is a pioneering vascular string technique. Before this invention of this technique, it's hard to, storing the technique. So he's opened the way the organ transplantation by this technique, but he is also famous making this Carrel's flask. He invented this Carrel's flask, and then he cultured the embryonic chicken heart in the Carrel's flask. So now you can see this is a glass, a culture dish, and with this like a cotton is clogged, and then no germs and no are coming into this culture dish. So you can keep the cell sterile and culture in more safe conditions. And then what he did is he cultured the embryonic chicken heart in this Carrel's flask more than 20 years. And if he found that the 20 years is longer than the chicken's normal lifespan, so he thought there is a senescence at that time, and because it's this, if you culture the cell, the cell is live longer than their lifespan. And then now we don't use this glass culture plate, and then now we use this plastic Carol's flask. We still use this materials, and then very useful. So this cap, there is filter in this cap, and then the air is coming freely, but it's any germs and other bacteria stuff cannot enter this flask. And then phase contrast microscope, also the great invention in cell culture. Then this Fritz Zernike, a Dutch physicist, he invented a phase contrast microscope. And then the beauty of this phase contrast microscope, you can observe the cell without staining, so you can observe the live cell. So before invention of this phase contrast microscope, you need to fix the cells and stain the cell, and then observe the cell. So you have to observe the dead cell, but before and after this phase contrast microscope inventions, you can observe the live cells. And then in this microscope have this like sliders and containing this like phase contrast ring. And then I'm not a physicist, I don't know how this optics works, but you can observe the cell. So this is a normal microscope imaging, and you don't see a clear cell shape or structure using a normal light microscope, because the normal light microscope, you just observe the light, it's through the cells, and cell is almost transparent. You cannot observe the clear cell shape. But if you use this phase contrast microscope, you can observe the cell shape without staining. So it's a great invention for cell culture. And cryospolage is identify the glycerol as a cryoprotectant. Well, this technique can be used to freeze the cells and thaw the cell. So you need to freeze and thaw the cells. So you can stop the cell culture when you keep it in the cell in a glycerol stock, and then you can just thaw when you want to do an experiment. And then Hayfrick found the cell is not the senescence, and then if you culture over and over, the cells start to die. So he called it's a Hayflick limit. So the cell is not a senescence. And then from this Hayflick limit, another researcher found the telomere. It's the capping gene of the chromosome, and then digestion of the telomere gene show the lifetime of their cells. And then, so this is the history of cell culture. So cell culture is not invented by one single researcher. It's many inventors are involved in this cell culture technique. And then next I'm going to talk about how to isolate the cell. And then there are two ways to isolate the cell for cell culture. One is primary cell culture, and you need a live animal and dissect tissues and then dissociate the cell using an enzyme like a trypsin, or sometimes use EDTA to just dissociate cells, and use this cell for to observe the cellular response. Then the good part of this primary cell culture is this cell itself the cellular response is very physiological. The bad part of the primary cell culture is the cell is not homogeneity. So multiple cell types are included in this primary cell culture. Even this liver cell, if you want to culture hepatocyte, but this liver contains the blood cells like endothelial cells, pericyte, macrophages. A lot of cells are dissociated after you can obtain after this dissociation. The cell populations are very multiple in primary cell culture, and they usually differentiate itself. Even hepatocytes have limited lifespan. So every time you do this cell culture, you need to kill this animal. So it's not the ethic. Okay. But another way to use this for cell culture is using a cell line. You can buy a cell line and then you can just culture the cell without killing animals. And how to develop this cell line is, so first you need to kill the animals to get the cells, and then, but don't use this cell directly. So then apply the random mutation and deliberate modifications and to let them have proliferation activity. So, and then, which means lets this cell to be a cancer and to acquire some more proliferation activity. So then if you use this, you can get the homogeneity of cells. This is a clone. So every single cells are same gene set. And then you can culture indefinite proliferation because it's cancer cell. And then it is very ethical because you don't need to kill animal every single experiment. And then bad parts of this immortalized cell line usage is the response is less physiological because this is a part of the cancer cells. So the normal physiological cells do not have proliferation activity, but this cell had become a cancer to get a mitosis. And then, so they use more energy for proliferation, and then it's kind of trade-off. So once the cell get the proliferation activity, they lost some physiological functions. So the response, the cellular response you observe, not always the real response that you see in the physiological condition. And then this is just a side story. So it's like 2018, I think I submit the paper using the cell line, but this paper, I don't know which paper, Journal of Endocrinology, is rejected my paper because I just use a single cell line experiment and then discuss the cellular response. And then this editor did not like just show the single cell line experiment. And no, recently it's hard to accept this the experiment using just single cell line. So just because as I said, the cell response is not always physiological. And then today's main topic, I'm going to talk about the three-dimensional cell culture. And then, so now you get to know how to get the cells, and then you start cell culture. And usual normal cell culture use a 2D cell culture like Carrel's flask, which is a culture of the cells in a flask. But today I'm going to talk about this 3D cell. And so this is the take-home message today's lecture, and it is the 3D and 2D cell culture is not identical. And so it's the 2D cell culture, a 3D cell culture is not just a stack of this 2D cell culture. Cellular response are different. And the most famous example of this, showing the difference between 2D and 3D cell culture is this embryonic stem cell. As you know, embryonic stem cell can differentiate into the multiple lineage like cardiomyocyte neurons and hepatocytes and then epithelial cells. And this is the ES cell culture in 2D, so in a dish. but you cannot directly induce this ES cell into this cardiomyocyte or hepatocyte from this using just a 2D cell culture. And you need to first incubate this ES cell in 3D environment and make this embryonic body. And this is a 3D culture, so cell aggregate. And then ES cell is the location of ES cell. It's more like in physiological context, and then you can induce these multiple lineage from this ES cell. And then there are several studies just compare the 2D and 3D cell. And this group who just culture smooth muscle cell line in 2D and 3D. And then this is the image of the cells and 2D cells and 3D cells. This one is probably the culture 3D in 3D and dissociate cells. And then stain actin showing the stress fiber, and the vinculin is showing the focal adhesion molecule. And if you look carefully, compare these two cells, and 2D cell is more elongated compared to 3D, and then in 2D more focal adhesion molecules expressions compared to 3D cells. And then this group also compared the DNA content and RNA contents, and then both contents are significantly decreased in 3D environment, which means the cell number is decreased when you culture in 3D. And then they check the cell growth inhibition marker P21, and then this is a proliferation marker P53, which is a cancer markers. And also collagens and MMP. MMP is our enzyme to digest collagens. And then what they found is they found that the cell number is the decrease when culture 3D. And then the reason is just it's in 3D, cell growth inhibition molecules is increased, not the cell proliferation is decreased. And also they found the collagen extracellular matrix is expression is increased in 3D, but this is not by the digestion of inhibition of digestion of collagen by MMP. And then also they found this the reduction of actin expression, which show the stress fiber markers. And then this is the beta-actin is our internal controls, and C-force and C-jones are transcription factor. Those expressions are more significantly increased in 3D environment. So you just culture same cell, but two different conditions, so you get the different expression genes. And then another group, they do almost the same study using a melanoma cells and then culture 2D and 3D. And this is cell populations over culture time. And as you can see, the cell growth is quick when culture in 2D cell environment, but the cell proliferation rate is very slow in 3D. And then this group also did DNA microarray, which kind of old technique now. People use RNA-seq to compare the gene expression. But briefly, let me explain how DNA microarrays work. And this is say normal and tumor, but it's say just 2D, 3D culture cell. And in this case, and then isolate the RNA, and then just dye this RNA with green from 2D, and then dye this RNA from 3D culture in red. And if you mix 1-to-1 ratio, and you get the yellow color when the gene expression in 2D and 3D is the same. But if the gene expression in 2D is more then 3D cell culture, you get green signals. And on the other hand, when the gene expression in 3D is more than 2D, you get the red signal. So if you compare the color, you can see what genes expressions are increased or decreased in this cassette. And then I skip these results, but this is sample results. So melanoma cell, they checked the 20,000 genes and found 173 genes expressions are altered by just change the culture condition from 2D to 3D. And the first group also did this microarray, and they checked the 9,600 genes, and they found almost like 100 genes expressions are altered by 3D cell culture. So from these results, about 1% of gene expressions are altered just by a culture context. And then when I summarize what type of genes are altered by 3D. So this is a list of the upregulation genes in 3D cell culture. So when you culture the cell in 3D, so P21, which is a cell growth inhibition markers, are upregulated, and then cell signaling molecules and then extracellular matrix, including collagen, fibrogenin, hyaluronic acid, are also factors are increased. On the other hand, here's a list of the downregulation gene in 3D culture, and one is this proliferation markers and phase phosphoproteins and cell division cell cycle 27, and then actin and rho, which is important protein to form a stress fiber, so cell stretching, and then vinculin. As I show in the previous slide, the focal adhesions molecules are decreased in 3D context. And then 99% genes are pretty much the same in both conditions. And then this vinculin also accelerate the focal adhesion kinase dephosphorylation. That's also promote the P21 expressions. So this cycle occurs in 3D context. And then here's a summary, how does 2D do? And so when you culture the cell in a dish, the cell only have lateral interactions, no multiple interactions, and then cells are only attached on the bottom. So only the extracellular matrix are on the bottom side. So the interactions are very limited. So then they lost the cellular polarity, and then they spread, and they are more squamous, and then spread to become make a stretch fibers, and then to be more stretched, and then that's induce the cell proliferation. So when you kill the cell in 2D cell culture, promote the redundant cell spreading, focal adhesion, and stress fiber formation, and then which result in the more proliferations. So the cell use more energy for proliferation rather than their original physiological functions. And then this is a summary of the 3Ds, and then cell have multiple interactions, more three-dimensional interactions, and then extracellular matrix is around all the cells, and then cell plurality is pretty even, and then this cell stop the cell proliferations, and then just mimic as they should be in the physiological environment. And then here's the several way to culture the cell in 3D. So I used to use this hanging drop method, and then just culture the cell in this drop on the culture distillate, and then you can just add like 1,000 cells or 10,000 cells per like 30 micro or 50 microliter of culture media, and then the cells are start to form a cell cluster at the tip of the drops, and then you can get the cell cluster and then see the several response. Another way is this suspension culture. You just like probably you don't know how to make a snowball, and because you're living in a tropical area, but like a snowball is like it's getting bigger and bigger, and then when you like rotate the balls, and same like physics like this, and then just suspend the cell, and then cell attach together, and then it's bigger and bigger. And you can also use this matrix scaffold. So like you like a spongy like extracellular matrix structures, and then keep the cell in this like structures and get the 3D. And these two way is you need a special equipment, and this matrix scaffold is pretty much expensive. And then this is very simple, and then you can get the same size of cell by controlling the cell number in each drops. So from now I'm just show only the data. So from now it's more like experimental work. Well, when I was done in Biomedical University about pituitary tissue formations. As you know, pituitary is produce many hormones, and then it's control the human physiology. And then anterior pituitary produce 6 types of hormones like growth hormone and ACTH, and then TSH, prolactin. Many hormone producing cells are located in anterior pituitary, but in anterior pituitary tissue, each cell are intermingled in these tissues, and it's very looks random, a mosaic distributions. And then in this tissue, there is a cell do not produce hormones. We call it follicular striate cells, which is a non-endocrine cell. Showing here, you can see you don't see any secretory granule in this site, in the cytoplasm. These black dots are secretory granules that contain the hormones. So these types are large granules imply the growth hormones. And then this type 1 is, I'm not sure, probably ACTH. So one cell produces one hormone except the LH and FSH. And then you can identify the cells by this electron microscopy. But again, you can also observe the cell do not produce hormones in this tissue. And then we want to know the function, what these cells do in anterior pituitary. And then these cells are form cell-to-cell interaction, homophilic. So FS2, FS follicular cell 2. Now we say FS cell, follicular cell as a FS cell for just it's more easy to say. And then this FS cell interact each other, and FSL to FSL to form a gap junctions and desmosome, and then form the network, cellular network within this anterior pituitary. So it is so that this FSL is like oscillate the function of hormone producing cell in the anterior pituitary. And also it is so that this is a stem cell and phagocyte, and then such as so on. And then the first one, what I did is the culture this anterior pituitary cell in 3D environment. So before I invented this cell culture technique, the people culture pituitary cell in only 2D. So I think to see more cellular response in more physiological context, we start the 3D cell culture. So this is a cell culture. So this is primary cell culture. So we kill the animals and dissect pituitary and dissociate cells and culture the cell in the drops and get the cell cluster at the end tip of these drops. And here's the cell aggregates. And then when I stain the FS cell in green, and you can see, and this is a 3D image using a confocal laser microscope. And you see these like cell-to-cell interaction even within this cell aggregates. This is the FSL in like live in tissue, so it's very comparable. The cellular locations and cellular formations are pretty much similar to what it should be in the in vivo pituitary. And then we can also confirm the presence of gap junction between the folic FSL to FSL. So this is FSLs here, and then this is very small, the projection of FSLs here. And we can see the gap junction here. So the molecule itself is also formed in this 3D context. And then a previous slide showed in 3D context, the extracellular matrix depositions are increased in 3D. And then I stain the 3D cell aggregate with a type 1 collagen and type 3 collagen, type 4 collagens, and laminin fibronectin, which are all extracellular matrix seen in the anterior pituitary, and you can see these depositions in between the cells. And the next experiment I did is just to see the function of FSL. We just split the cell into like FSL and other pituitary cell type by using a cell sorter. And then in this experiment, I use transgenic mice that express green fluorescent protein only in follicular stem cells. So you can separate the green cell and non-green cell by cell sorter and then combine together for a 3D culture. One is just a normal FSL population, 5%. And the other is just without FSL, just culture the cell without FSL and compare what's the difference between these two cell clusters. And what I found is this is a presence of FSL and this is the absence of FSL. The cell cluster is more big in without FSL. And this is more compact. We culture the same number of cells in 3D context, but the cell shape are very different. So this is for more like round, and this is like irregular shape. And now this is at the bottom image are electron microscope image, and then we can observe this is a collagen fiber. And you can see like striations of the fibers. This is a very typical collagen pattern in electron microscope. So we can observe the collagen, but we didn't observe any collagen deposition in this cell culture without FSL. And then just in case we check the gene expression of collagen type 1 and collagen type 3, and then expression levels are significantly decreased when cultured without FSL. So without FSL, the anterior pituitary cells stop producing extracellular matrix collagens. And then this is the staining of collagen and with FSL and without FSL, type 1 collagen and type 3 collagen. And then as it's obvious, you don't see any deposition of collagen in this cluster when cultured without FSL. And then this collagen is produced in the parasite in anterior pituitary, which is already published in Dr. Fujiwara. Now he is in Kanagawa University. And the parasite is a type of stellate cells of microvasculature. This is a capillary endothelial cells, and this parasite support this capillary, and then function of parasite is control the vascular tone and also produce an extracellular matrix for these endothelial cells. So we see the decrease in collagen productions, and we think that this FSL, there should be an interaction of FSL and parasite. And then now we check the gene marker for parasite, which is named desmin. And then again, the desmin expression also decreases when you culture the cell without FSL. And then parasite, presence of parasite is pretty much decreased. So red cell is parasite. So more parasites are seen in 3D when cultured with FSL, and then less parasite when cultured without FSL. And then collagen production is affected by transforming growth factor beta, and then which are produced only in FSLs. And then I think that this FSL, the TGF-beta 2 from FSLs are important for a production of collagen from parasite. And then this is gene expression level. So when you add more TGF-beta, you get more collagen expressions. So to summarize this FSLs are produce a TGF-beta as a paracrine fashions, and then this TGF-beta parasite receive this TGF-beta to produce a collagen, but this FSL needs this collagen for to form a cellular interaction in the anterior pituitary. So we found that there are two cell interaction using this 3D cell culture. It's hard to find this stuff in 2D cell. And then now I changed the topic about the laminin, which is another extracellular matrix component of vasculature, which is stained in red, and green is FSL. And then as you can see, FSL always attached to the basement membrane component laminin. And then laminin, here's a vasculature, and here's a vasculature, and then basement membrane is surrounded the vasculature, and then FSL always attached to the basement membrane to just hold themselves in this pituitary tissue. And then first I checked which cells produce laminin. And then I found two cell types produce laminin. One is LH cell, which produce luteinizing hormones, and another one is vascular endothelial cells. Those two types produce laminin in anterior pituitary. And then this is a paper from Ramadhani, my Indonesian student, and almost more than 10 years ago at Biomedical University. And then again, I changed the proportion of FSL like 0% to 20% and stained the laminin. And then I found that 0% FSL laminin is only seen in the cytoplasm. But when you increase the proportion of FSL, we don't see such the cells, red cells, and then we see the deposition of laminin. So this is a number of laminin positive cell like this is significantly decreased when you increase the proportion of FSLs. So from this study, I found that FSL also interact with L8 cells, luteinizing hormone cell, which produce a laminin, and then we don't know what paracrine factor are involved to let the L8 cell to produce laminin, but we found a new interaction, these two cells, and then this laminin bear through this laminin. And laminin is also important for FSL because FSL use this laminin for anchoring. So then this is the last slide and then this take-home message is just if we want to see the simple cell response, it's a 2D is just easy, but if we want to see more physiological cell-to-cell interactions and cell-to-extracellular matrix communications, and then you better use 3D cell culture because the cellular response are different in between 2D and 3D. And this is the end of my lectures. Thanks for listening. Thank you. I'm just back to Jimmy-san.
Okay, thank you, Tsukada sensei.
Mm-hmm.
Very clear, and it kind of remind me what I did at the DJ when I was there. Yeah, I learned a lot in the laboratory. Okay, now we go to the Q&A session. I really encourage the students to ask Tsukada sensei about the topics of 2D and 3D culture, and also maybe to clarify some things that you don't understand. Please feel free. Okay, we have one student raising hand, Yuli Dwi Haryani.
Hello, is my voice audible?
Yes, it's audible, Yuli. What's your question?
Hi.
Okay. Hello, Mr. Takehiro. My name is Yuli. Thank you for the comprehensive explanation. I would like to appreciate Biomedical Science teacher and team to conduct this event. I want to ask you a question, Mr. Takehiro. You earlier mentioned that 3D culture mimics the in vivo environment. Does that mean that in the future we could rely solely into in vitro research for various studies, or are there still challenges associated with this approach? The second question is also I want to hear your opinion. Does using 2D cell culture make research the result of the research less valid? Thank you for the opportunities.
Okay, I couldn't hear the first question clearly. Can you say it again?
Okay, you said that in the presentation, 3D culture mimics the in vivo environment. Does that mean that in the future we could solely depend on in vitro research for various studies, or are there still challenges associated with this approach?
Okay, the first question is just 3D experiment, and then you think it's value compared to in vivo research. So there are two. It depends on what you want to see, and then if the final output comes from the whole interaction in the body, like as I said, the nervous system, and also blood pressures, and then a lot of things are involved in just one cellular response. And then that's probably the final goal, to figure out what's the gene or what the cells do in this animal, the whole animal individuals. And then if you want to see this a very super comprehensive the works, so and just better use the in vivo study, but you don't get the clear answer by that because it's too much complicated. And then, but when you use a 3D cell culture, you can simplify this just the cell to E cell interactions without any effect of nervous interactions or immune cell interactions or hormonal interactions because it's very artificial environment. So if you want to see, your purpose is just see the cell to cell simple interactions, what's happened in the cell to cell or cell to ECM, and then I recommend to use a 3D cell culture system. And then the second question is I think still 2D culture is more value. And so if you do a cell culture, just please do a cell culture. But again, it depends on your purpose and what you want to know. And then you pick this sometimes 2D because it's easy and simple. And then if you check more like cell to cell interaction, the 3D is the best way. But in 3D cell, it's hard to observe in the microscope because you don't see the inside of the cell. So you need a special like microscope, confocal laser microscope is required, which is pretty much pricey. And if your facility do not have the laser microscope, you cannot, you can culture 3D, but you cannot observe what's happening this. But like gene expressions, I think you can do in your, if you have a PCR, you just culture 2D and 3D and then compare these two expressions. So.
Does that answer your question, Yuli? Or do you want to?
Hello, Mr. Takehiro.
Thanks.
We call him Dr. Tsukada.
It doesn't matter.
Tsukada Sensei. Yeah, is there any other questions, students? It's a good opportunity for you to discuss with international scientists. Yeah, it's a rare opportunity for you. Come on, take this a chance. Okay, Ati, you have any questions for Dr. Tsukada?
Oh, yes. Before, thank you, Mr. Tsukada. Hajimemashite. Watashi no namae wa Rhea desu. Watashi karakemashita. Watashi wa Shion desu from a new student from Biomedical Science 2025. Medical Faculty of Medicine, Andalas University. And I have one simple question. Actually, I just wanted to say for you, your presentation today is sugoi and kakoi. Because I'm very interested with all about Japan. I think like people, language, and all about Japan. I am very interested. So my question just can you give us tips and trick and motivation for you student, our new student to continue to innovate, especially in research up to international level, because a new student in here. So can you give our motivation to continue research? And I think I'm very interested for your presentation because your is sugoi.
Okay,
Maybe
yeah.
and the last dessert yoroshiku onegaishimasu. Arigatou gozaimasu, Mr. Tsukada.
Okay. Hi, thanks, Ati-san. So for a young researcher to how to keep their motivation more in science is just open mind is just most important parts. And it's you just focusing on just one simple things, and then you cannot see the other parts. But there are many things happens in the world, and then like even in this small body, just simple cell, we don't see any more. We see some function, but most of function of just single cell is very mysterious. And then just open mind and then just be curious any type of like phenomenons, why this cell do this or why the animal to do this. And if you just had your curiosity is just a most important part to just keep the students' motivation is high.
Okay, thank you, Sensei. So Ati, just keep on being curious, keep an open mind. That's a message. Any other
And
questions?
one more thing is just exposing yourself to like more higher levels and like more just expose yourself more internationals. That's also important, you know. Even in just Japan, it's not very, very small communities, but its science community is more large and worldwide. And then just open mind and just feel free to just ask any. Now you have many tools like Zooms and emails, and you can communicate. Many people from any countries, most of the countries, not any country, but most of the countries, and then you have more opportunity than I had before, I guess.
Yeah, Sensei, that's right. It's amazing that how we can connect like this. I didn't imagine we can do this when I was there, right?
Right.
Yeah, the world is getting smaller, and so the message is, students, you have to keep exposing yourself to high level research. Just get familiar with that, and I would like to invite more questions, please. We only have 2 so far. Come on. Oh, we have Fanny Morina. Okay, Fanny, what's the question?
Okay, hello, Mr. Tsukada. I want to ask some question. I think I want to ask you, in what cases do we prefer the 3D cell culture instead of animal models? Because I think in 3D cell culture, the ECM is crucial because it allows cell to behave more like they do in vivo, supporting more realistic studies of cell behavior, the interaction of the cell. So what cases do we prefer to use 3D cell culture? And can we also examine the extracellular matrix or ECM in animal model research?
So what case? So again, just if you want to see the cell-to-cell interaction in more artificial conditions, the 3D is the good way to check the cellular response, and then but it's basically from the curiosity of what's happened in the animal. This is the first question, right? So you need to see the phenomenon in the whole animals, and then you have questions. But it's hard to address the answer using the in vivo study, like a whole animal study. Then you need to just break down the each parts, and then if you think it's a cell-to-cell interaction or cell-to-ECM interactions are critical or can be explained that phenomena seen in the whole animal, then you can use the 3D cell culture. Don't start the 3D culture first, right? Because it's just a mimicking the cells in vivo, right? So then the first important part is just what is curious and what's the interesting phenomena you see in whole animals. Okay.
Is that clear, Fanny? Is your question answered?
Okay.
Didn't I answer?
Do you have any idea, Fanny? What kind of research you want to do? She's still in her first semester.
Yeah.
Uh-huh.
So for you, maybe you're still formulating the questions, and I want you to know that Tsukada Sensei has master student in your laboratory right now. Is that
Yeah, I have two master students.
two master student, and
Mhm.
now you're working on eel physiology, right?
Right.
And the only laboratory in the world working in the eel physiology.
I don't think so. There are many the laboratory they're working
Okay.
on the EO study, but I'm combining the EO study to more medical study. So probably this is the only one laboratory. So EO to medical school, from EO to medical research.
Wow.
That's
So
probably
that's
the only one laboratory in the world.
yeah. I
So
think
most
you can like promote to my students. Maybe they want to pursue that because they have some have medical backgrounds
mm-hmm.
and how
Right.
eel relates. Yeah.
Mm-hmm. That's yeah, that's a good idea. And then
Yeah. Is
I
there
need
any
more students.
opportunity for my students like to do short training? Maybe they can learn what some techniques in your laboratory if they're
Yeah.
relevant.
Yeah. No, I'm always welcome your students, but budget-wise, if
Yeah.
your budget is come to your university, it's no problem.
Yeah.
But it's hard to. We need to write the JSPS like fellows, and then it's we can formally accept because financial wise is the most the key. But if
Yeah, we have so many opportunities actually, grants we can apply for, Sensei.
yeah,
I'm
then
looking for avenues to send my students so they get international experience. Yeah, I'm excited to explore this in the future. Is there any other questions, students? If there's no more questions, oh, we have Reski. Okay, ask away, Reski.
Okay, thank you for the opportunity. So my question is, what are the main technical challenge and limitation in standardizing 3D cells culture protocol for reproducibility across different laboratories? Because yeah, this grow so many challenges I see in this reproducibility in 3D cells. So what is the main technical challenge, issues, and limitation to reproducibility across different laboratories? Thank you.
it's what's the limitation of what 3D culture or
Yeah,
It's not
what is
very clear, the audio, Reski.
okay? Is my voice audible right now?
just a moment, I increase the volume just now. Yeah, can you?
Okay, thank you.
And now I can hear.
Based on what you have present to us, I want to ask some question. It is about the limitations and a future challenge in standardizing 3D cells culture protocols for reproducibility across different laboratories. I think this I want to know about the limitations and the technical challenge.
So this 3D cell culture is a good technique to show the cell to cell response, but it's just one of the technique to confirm the cell to cell interactions. So only the data from 3D cell is not the perfect. So you need another experiment to confirm the result from 3D cell culture, and then you need more comprehensive view and then comprehensive experiment to just confirm what's happening in the cells. So that's limitation. Not only a 3D cells, but any type of experiment have limitations, and then to solve the limitation, to just get rid of the limitation, we need another technique or another way to check to confirm the cell response or just physiology cell response. Did I answer correct? Chimi-san, is Chimi-san here? I cannot see Miyawati-san. Probably the computer is crashed or just wait. Does anyone know why Emerson is absent now? It's just computer problems or Emerson.
Oh,
Chimi-san.
I just have a connection problem.
So now I can
know.
see you now.
Yeah. And so we still have 15 minutes for Q&A. Students, do you have any other questions for Tsukada Sensei? Oh, we have Muhammad Abi, Gofari, and Uswatul Hasanah. First, Abi.
Okay, thank you for the James. Okay, thank you for the Dr. Takehiro. I'm gonna ask you something like concern my research. My research is about biomarkers in detecting endothelial dysfunction. As we know, in the research, many biomarkers have different pathways. Can we combine these biomarkers in the cell, or like can we detect in cell culture and then we combine of these biomarkers? Okay, thank you.
So I couldn't find the first the part, and then what is you're doing now? Is the biomarker?
Yes, biomarker in the endothelial dysfunction.
In endothelial cell functions.
I have two biomarkers and others pathway, but but I want to combine the two biomarker to detecting in endothelial dysfunction.
So you want to detect the two biomarkers in endothelial cells?
Yes.
And then if you what biomarkers you want to see, like proteins or like
a protein, yes.
Proteins. So do you have antibodies?
Yes, the antibody in the PCR or like ELISA.
ELISA. So you see the gene expression by PCR, and then the two marker genes are expressed.
Right? And then we combine it. Can I the research can?
Oh, you want
what
to
combine?
What
do you want to know, Abi? Okay.
The application of 3D in
your
research?
Yes, application the research.
Okay, so now you're working on endothelial cells, and it's also the cellular localization is very important. And then the endothelial cells requires the parasite that I show in my slide that produce a collagen. So simply you use this. Are you using a cell culture or like live animals? Which?
Yes, like animal.
Live animals. Yes. So as long as you working on the live animals, it's a set of the vasculatures are fine. So it's you don't you're right. But if you want to combine 3D cell culture, you just culture like epithelial endothelial cells and also other vascular cells type like parasites, and then you culture. So culture two cells and see the cellular interactions, and it's doable in 3D, but it doesn't necessary to culture in 3D, but you can culture just two simple cells and then to see the interaction of these cells.
Can I implement to vascular endothelial, like in vitro maybe?
Yeah, it's doable to culture the endothelial cell in vivo, in vitro, while even in a 3D cell culture is doable. And then you can make a tubular formation in cultures. So you can culture the cell and then see the interactions. But probably you need a special, like the matrix scaffold stuff to culture 3D, because like make a 3D like a scaffold of the tubule and then culture endothelial cells on that scaffold, and then the cell start to form a tubule, and then you can culture it, but probably it's technically very difficult to culture it, and then you need special equipment for that.
Okay, thank you, Mr. Thank you very much.
That's the challenge, Abi. Okay, and next, Uswatul Hasanah, you have a question for Dr. Tsukada? Uswatul Hasanah?
Yes,
Still here?
thank you.
Okay,
Thank
please.
you, Mrs. Simi. Hello, Mr. Tsukada.
Hello.
Yeah, it's my voice is clear. It's my voice audible.
I can hear you.
Yeah. First, actually, I'm in the first semester in biomedics magister, but I have interest in nutritional experiment. And my question is, can we use 3D cell culture to see interaction between cell target and nutritional material or some extract of some food? And what kind of the difficulties of bias when we use 3D cell culture to this nutritional experiment? Thank you.
So nutrition experiments. So if you use like 3D and nutrition experiments is kind of, so not, so those either if you do a 2D cell culture or 3D cell culture, you can modify the cell culture conditions, and you can add or just like no nutrition and see the server response. So it's, if you want to see the effect of some like nutrients you are interested, and on the cells, it's the 2D, it's 2D or 3D cell culture you can use. But it depends but it's, if only like nutrients only in the circulations in the bloodstreams, the cells are probably okay. Yeah, then probably you can use the cell culture, just see the effect of nutrients on the cells, and then see the expression levels. But it's 3D cell culture is not always necessary then in your experiment, I guess.
It's not always necessary. It really depends what you want to see, what the effects of the nutrients on what cells, what's the function of nutrients. Yeah, you really should like get more specifics on your research questions. Yeah, Uswato Hasana. Okay, thank you for the questions. And is there any other questions or something you want to clarify? No more questions? I think we have a special attendee today, Dr. Reza. Dr. Reza, like, want to say hi to Dr. Tsukada. So, Dr. Reza, you want to say hi?
Yes, of course.
Okay.
Yes. Hello.
Yes,
Tsukada
please.
Sensei.
Okay.
Hello, Reza-san.
Hello. I'm very happy to see you again. Yeah. Currently, I heard about your lecture, and yeah, but too bad I'm not in the Wales. You know, I'm working around sometimes in the field, so I just have to and sometimes I have to come to my car to listen to your audio, to your lecture. So how is life? It's been a long time. I haven't heard any news. Yeah, last
I'm
time I
doing great.
remember. Yeah, last time I remember, it's 1 year ago. I had your lecture also, right?
Right.
It's about 1 year.
Almost 1 year ago.
Yeah.
Mhm,
Maybe
I'm
you
very
will be
happy
our
to see.
regular guest and say.
Mhm.
I hope you will be our regular guest. Yeah, it should be
Yeah,
fun.
yeah. I want to go.
Oh,
Yeah,
be
yeah.
happy to.
Hopefully.
Yeah.
Yeah, wait for me in
Okay.
Japan. Yeah, we'll see each other.
Yeah, we'll see.
Okay.
Good luck
Okay.
then.
Thank you, Dr.
Nice to
Reza.
meet you.
Thanks,
Bye-bye.
Reza-san.
Thank you. You're welcome, Sensei.
Okay. And is there any other questions, students? If there is, oh, we also have some lecturer here. And Sensei, for the first time this year, my graduate school, we have an international student. So maybe next year I want to send my student to Japan, hopefully to your laboratory for
Mhm.
like a short-term training.
Okay.
Yeah, we'll see that.
welcome.
We'll discuss about that. Yeah,
Uh-huh.
hopefully.
Yeah.
Okay, now, Sensei, I think I would like to conclude this session because there's no more questions.
Okay.
Yeah, and thank you so much for sharing with us. It's been a
Thank
pleasure for us.
you very much.
Thank you very much. Everyone, please give a warm applause for Dr. Tsukada. Let's give him.
Thank you.
Okay. Thank you, Sensei.
Bye.
And please stay in the room, everyone, because you need to fill the post test. And Sensei, if you have something to do, you can leave the room, but I will communicate further.
Uh-huh. I have another meeting from 1 o'clock, and then I
Yeah.
need to leave the
Yeah.
Zoom room. I'm sorry, but I'm very happy to share the time with you and your students. And then hopefully next time we can see, and then you can send your students to my laboratory. I'm very happy. Yep. Okay. So
Okay, thank you,
you very much.
Sensei.
And then
Have a
yeah,
good
I'm gonna leave
day.
now. Hi. Have a good day. Bye.
Have a good day,
Bye.
Tsukada Sensei. Bye-bye. See you.
Hi. Nice to.
Oke, nah silakan diisi post testnya ya. Di-share lagi linknya, Dea. Silakan mahasiswa Bapak Ibu 5 menit saja ya. Nah, kalau tadi menyimak kuliahnya pasti post testnya benar semua ini. Kemudian ada juga umpan
Link,
baliknya untuk
linknya belum
guest
di-share, Dr.
lecture.
Oke.
Oke, di kolom chat kita share link post test juga. Silakan diisi ya Bapak Ibu, mahasiswa semua, umpan baliknya juga supaya dapat sertifikat, dan umpan balik ini untuk perbaikan ya, perbaikan guest lecture ke depannya. Silakan dicoba lagi barusan ya, dibuka untuk accept response, post test. Maaf Bapak Ibu ya, bukan tidak menerima jawaban, belum dibuka tadi, belum diklik. Maaf ya, silahkan diulang lagi. Bagi mahasiswa dan Bapak Ibu yang memerlukan materi guest lecture tadi akan kita share di grup peserta. Baik, kita sudah terima respon post-test ya. Link post-test tetap akan kami buka 30 menit ke depan. Kita tutup saja acara guest lecture pagi menjelang siang ini. Untuk sertifikat akan dikirimkan ke WA atau email Bapak Ibu mahasiswa semua kurang lebih dalam waktu 2 minggu ke depan. Terima kasih atas perhatiannya. Mohon maaf atas segala kekurangan kami dalam menyelenggarakan guest lecture ini. Dengan ini kami tutup acara guest lecture. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.